Hello everyone. Today we are going to study genetics part 7, and before starting with the actual topic of today's lecture, we will first solve some questions based on genetics, monohybrid cross. So if suppose you have the first question as, so we will discuss questions first. This is question solving class basically. Let's solve 2-3 questions based on genetics. So first question, suppose if the question is what what will be the appearance of F1 and F2 generation, F2 F1 and F2 generation when a pure tall pea plant is crossed with a pure dwarf plant. Okay, so this is the question. So what will be the appearance of F1 and F2 generation when a pure tall pea plant? So they are saying what? Pure tall pea plant. Okay, this is first, is crossed with a pure dwarf. So this is the question. Let us take pure pea plant. Pure pea plant means it will be like this. Pure means homozygous tall. So here you will first show the genotype and you have to mention the phenotype in the brackets. I'm just solving the question for now. We have to follow the exact procedure the way we follow during solving monohybrid cross. So if I take homozygous tall, that shows pure pea plant. If I take pure dwarf plant, then it shows homozygous dwarf. So here, since these two alleles are similar, so what we will do, we will take only one allele as gamete. So let's take capital T as gamete and let's take small t as gamete. So here there will be fusion of gametes, then you will get capital T, small t. So this will be F1 generation where all the plants are tall. So you will write all are tall. All plants are tall. You can mention your plants also if you want. Okay. I don't want to write much, so I'm just writing this much. This is F1 generation. Now what we do for F2 generation, if we want to know the possibilities of F2 generation, then we will do here selfing. Okay, selfing means self-pollination. I will continue on the next page. So, okay, where is my pointer? So capital T, small t, cross with capital T, small t, as we are doing selfing here. So what you will get? Here we will get two gametes. We will first do gamete formation. So this capital T will separate, small t will separate, and again here capital T will separate, small t will separate. So as I had taught how to cross, how to take the possibility, same way I have to do 1 with 3, 1 with 4, 2 with 3, and 2 with 4. You have to show the arrows. Okay, and then what you have to do, you have to write your capital T, capital T, capital T, small t, then you have to write your capital T, small t, and small t, small t. Okay, so this is the first. You will get your which generation. Here you will get generation as F2 generation. You will get which generation? You will get F2 generation. So you got F1, you got F2 generation. So the question was asking that what will be the appearance when it is known as appearance of F1, F2 means they are asking what? They are asking you. Appearance means they are asking phenotype. What they are asking you? Phenotype. So here I told you that all will be tall, so all F1 will be tall, and for F2 there are possibilities of 3 being tall, okay, and 1 being dwarf. Understood? So there are, I told you that what are the possibility. So here these 3 are tall as per the phenotype, and this will be dwarf. So the ratio comes as 3/4 is to 1/4, which is ultimately 3:1. So this is phenotypic ratio. Nobody is asking you phenotypic ratio. Just I'm telling you that what is the phenotypic ratio, just for the sake of knowledge. So this was our first question for today. I hope this is clear. You can take screenshots. You can write it down by pausing the video. I will move towards the second question. Now let's take second question as, now suppose I'm dictating the question here. Suppose the instead of pure tall pea plant and pure dwarf plant, the question had been this way, that a heterozygous pea plant is crossed. Okay, this plant is crossed with which plant? Heterozygous tall. Okay, heterozygous, I will write it here, tall. Heterozygous tall pea plant is crossed with dwarf plant. If this is the scenario, okay, I have written, you have to write your cross. Okay, you have to frame the question, same question, just you have to change the words. Then what will be the possibility? So what we will do, we will take first capital T, small t, as this is heterozygous and this is dwarf, so we will cross it with the dwarf genotype. So it's capital small t. So here you have to take how many gametes? Here you will have 2, capital T and small t. And here, since these two are similar, you can take both gametes also here, no problem. You can consider it here. Okay, so this will be capital T crossed with this capital T. Even if you don't take these two, then also it is fine. If you take only one, I will show both ways. No, there will be no such change. Okay, so here you get this twice. You have to show the arrows. Okay, so what you will get here, capital T, small t. You will again get capital T, small t. You will get small t, small t, small t, small t. So can I say that there is 50% possibility? Okay, there is how many percent possibility? 50% possibility is there to get tall plants, and another 50% possibility is there for dwarf plants. So 50% possibility to get tall plants, okay, and another 50% possibility to get dwarf plants. Okay, now I will show the second how you can do. Now suppose you are not separating these two gametes. Okay, suppose since these two are similar, you have taken only one time. So I will write the same thing. Here I am taking two gametes, capital T and small t. Here just I am taking as small t, sorry, since these two gametes are similar. So let's solve. Let's take capital T with small t, small t with small t, and we will show the arrows. Okay, here it is fusion of gametes. These are gametes formation. This is fertilization. Okay, same what I had taught you during monohybrid crops. So this is capital T, small t. This is small t, small t. Okay. So if you can see here, what is the possibility? Again, if you see that the possibility is that 50% is tall and 50% is dwarf. Okay, so here also you will write same 50% tall plants, and here also this is 50% dwarf plants. Okay, so this is about the question number 2 which we did here. Okay, I hope you understood this question. So these are the very simple question which I had taken. This is question number 1. Okay, then I took question number 2. Understood? If you are you understood till here, then we can move forward. Okay, by taking one more question based on this. So now we have taken it as heterozygous with dwarf. We have also done heterozygous with heterozygous. Okay, and this was heterozygous with tall. And the thing which we had done here, this is homozygous with dwarf. So 3 things we have studied here. Okay. Next we can say that question can be asked this way, that what is the what should be the genotypes of the plant. Okay, reverse question. What should be the genotypes of the plant in such a way that we should get 50% tall and 50% dwarf plants. So for this 50% tall and 50% 50 for this 50-50 person if you want. So what you have to do, the genotype should be heterozygous, tall and dwarf. Okay, not going into the much details of the problems, we will move step by step. Okay, so for now this is the question which I have taken. I hope these 2 questions were like easy. Now I will take one more question which is based on the third question we will move, which is based on this only monohybrid cross, but this cross is not done in it is not of pea plant. We will take off pig. Okay, in pigs. So in pigs what happened, that the question is saying that in pigs cross was done, and there was one allele capital B, and this allele was giving the the effect as black coat color. Okay, black coat color. Okay, if you want I will read the question once. In pig, dominant allele capital B produces black coat color, and its recessive allele small b produces white color. Okay, so capital B this is one allele which is producing black coat color, and the small b is producing which color? It is producing white color, white coat color. Coat color means skin color of that pig. Okay, coat color. Like what will be his skin color, we can say, coat. So they are saying that this capital B is nothing but this they have already mentioned in the question that this is dominant. Okay, and they have also mentioned that this small b is recessive. Now what the question is saying further, what will be the probable genotype of offspring when a cross is made between individuals heterozygous for the color? What they are saying that what is the probability? Okay, they are saying to you that what is the probability when two heterozygous organisms are crossed. Heterozygous means capital B, small b. If two heterozygous organisms are crossed, then what is the probability? So let's first write the gametes. So one gamete is capital B, second gamete is small b. Here also same you will get the gametes. Then you have to simply cross. Okay, 1 with 3, 1 with 4, 2 with 3, and 2 with 4. You have to put the arrows, okay, this way. Okay, and what we will write here? What are the possibilities we are getting? Capital B, then capital B, small b, then capital B, small b, then small b, small b. I hope you understood that how we got this. We got this by 1, 3. If you cross this way, 1, 3. And if you take, how 1, 3? If we number as 1, 2, 3, 4. So if 1 with 3 it comes, then you get capital B. When it is 1 with 4, you get capital B, small b. When it is 2 with 3, you get capital B, small b. When it is 2 with 4, you get small b, small b. Okay, so they were asking what is the probability. So if you can see that capital B, you have got this one time is to, you have got 2 times capital B, small b is to, you have got 1 time small b, small b. Now this capital B, this capital B is nothing but it will give you black coat color. Okay, again you have this capital B, small b as black coat color, and this also as black coat color, and you have only one possibility for white coat color. So the phenotypic ratio will be 3:1, means black coat color is to white coat color. Okay, so this is your phenotypic ratio, and you can see that I have written your genotypic ratio 1:2:1. This is your genotypic ratio. Okay, so the maximum amount, like you can say maximum number of the pigs will have black coat color, and only less number of pigs will have white coat color. So this is the third question which I wanted to take for today's class. Further, we will proceed for the next topic. What is Punnett Square? Okay, what is Punnett Square? Now we will consider this, that what is Punnett Square. So there was a scientist which gave a Punnett Square. Okay, what it gave? That scientist gave a representation method. Okay, that is known as Punnett Square, or we can say it as checker box, or we say as checkerboard. Okay, so it is a representation method. Okay, it shows you the permutation and combination. Now you have studied permutation and combination. These are the possibilities, like you calculate in mathematics, like what is permutation, what will be the possibilities whenever you have to calculate, that is permutation and combination in your you have studied this in mathematics 12th standard. If not, then you will study it. Okay, so this representations shows the possibility that how many offspring you will get. Okay, means which gametes will fuse after fusion of gametes, means after fertilization, means what I am saying, like how many gametes will fuse. Okay, which gametes will, okay, obviously two gametes will fuse, but which gametes will fuse. Okay, which gametes will fuse. Then after fusion means what? After fertilization. After fertilization, what you will, like, what you will land up, means which types of organisms you will get. So this all you can see in the diagrammatic representation. Okay, you can see, you have to make a square, like let us consider that in genotypic ratio of monohybrid cross. Okay, which cross we are taking? We are considering monohybrid cross. So the ratio was 1:2:1, and for phenotypic it was 3:1. Okay, this is for what? This is for your monohybrid cross. Okay, let us see monohybrid cross once again. We had taken capital T, this is homozygous tall, we crossed with homozygous trough. So since these two alleles were same, we took capital T. Since these two alleles were the same, we took small t. The fusion of gametes occurred, you got capital T, small t. This we told as this is the first F1 generation. Then we had to do what? We had to do selfie. Then we did selfie. You took capital T, small t crossed with capital T, small t. Okay. Then what you did, you made two gametes here also, you made two gametes here also. Right? Then you wrote what? You took it as capital T, small t. You took it as capital T, small t. Then what? I will show it here. Okay. Then you took what? You did this capital T with capital T, capital T with small t with capital T, small t with small t. So you got capital T, you got capital T, small t, capital T, small t, small t, small t. This was your, which, this was F1, okay, and this is your F2 generation. So can you see that here, which gametes we have taken? We have considered gametes as capital T, small t, capital T, small t. So what you have to do, you have to make a square. So first of all, let us make a decent square. So you can use ruler and all. Use neatly, because during your board exams you have to show it. Okay, so you should be like, you should be doing it very properly. Use your pens and everything, like pencil. Now suppose, like, suppose this is a square which we are taking. Okay, so I am trying to give you a proper idea. Okay, so I will make the table. and this I will go here. Okay, so I have taken here one table. Okay, so let's make this table little bit smaller. Now what you have to do, see, okay, now I think this table is perfect. So I will zoom this table. Okay, let us understand what we have to do. So here you can see here. Okay, I will zoom. Okay, zoom out. See, we have taken here which all gametes. So you can see here that which all gametes we have taken. You have your you can see that you have this these all gametes, capital T, small t. Okay, this is for mother, this is for father. Okay, we had done this in our previous class also. Okay, if you want we can see that also. We had done this in our previous class. Let us move there. If you want to see our previous class, what we had actually done. Okay, wait, give me a second. See, this was our previous class. Okay, and in our previous class we had done this. See, we had done this in our previous class. If you can see that I had taken here also female, male. Okay, and then if you move to the next slide. Okay, let's see this. Here it was. See, we have considered here. We had considered here this female and male. Same what we are studying in today's class also. Okay, so here what I have written. Okay, see, can you see that I have written here also that this is female. Okay, I'm just showing you that we have studied this already. If not, then do watch all the lectures of, all the lectures of this. Yes, do watch it, all the lectures of genetics. Okay, so this is female and this is male. So what we have to take, these are the gametes. Okay, these are what? Nothing but gametes. So I will come here to my square and I will write here female gametes this side. Okay, downwards. And this side I will write male gametes. Okay. What we will put here? We will write here female gametes and this side we will use what? Male gametes. Okay. If you will see that in the previous slide, I had shown that male gametes were capital T. Okay, I will put here capital T as male gamete. Okay. See, capital T as male gamete. Okay. This is capital T and this is small t. And if you will take this, so here also you have what? You have capital T and small t for female. In the previous slide, I will show it again. So what you have to do? You have to take this male gamete of the first column, and you have to take the female gamete of this column, this side of the. See, I am explaining. See, you have to take this um, this gamete of male. You have to fuse it with the first gamete of the female, so it will come here. Okay, so you have to take this gamete. Okay, you have to take this gamete and you have to fuse with this gamete and you will get this. Okay, so this gamete, which gamete you will take? This gamete. Take this gamete. Okay, fuse it with this gamete. So what it will be? Capital T fuses with capital T. Pink color capital T came from the male gamete and blue color capital T came from the female gamete. Let's move further. Here also again write capital T which came from the male gamete. Okay, male gamete it is, and small t from female gamete. Again you take, okay, small t of male gamete and capital T of female gamete. Okay, again you take what? Small t of female gamete and small t of male gamete. So if you can see that capital T gives what? Capital T means what? It is for tallness, tall height. Okay, even this is for tall height. Even heterozygous, this is heterozygous tall both. And if you want to say that which is homozygous dwarf, like what is homozygous dwarf? Like this is dwarf height, means short height. Okay, so this way if you will see, you will count. So 1 plus 1 plus 1, 3 are tall height. Okay, what are 3? You will get 3 as tall height and you will get 1 as dwarf height. What you are getting? 3 as tall height and 1 as dwarf height. You can also get 1 as capital T. You are getting 2 as capital T small t. You are getting 1 as small t. Okay, this is the thing which I have shown here also. So this is about Punnett square representation. You can show that what is the fusion and all, everything, what is the possibility and all. You can explain it by showing this particular representation. Okay, so I hope that this topic of Punnett square is understandable to you. I have drawn Punnett square for only monohybrid cross, since we have performed only monohybrid cross. Okay, I will zoom again. Just see how to represent. Okay. See, I have zoomed it. So this is about Punnett square. This is Punnett square for monohybrid cross. And I hope that you have understood this. Okay. So this is Punnett square. Punnett square for which cross? For monohybrid cross. Okay, Punnett Square, or you can say write checker board for monohybrid cross. Okay, so I hope that this is understood to you. Okay, so for today's class we will stop here, but in the next class we will study about, okay, in the next class we will study about the back cross and the test cross. Okay, so we will understand about what in the next class, whenever it will be, you will come to know about back cross. Okay, then you will understand test cross, and you will also understand dihybrid cross. Okay, so for this, stay tuned and keep learning, keep revising, revise daily. Okay, study the concept here, revise your state board NCRTs for your NEET, state board, and all the examinations which you have to face in the upcoming time. So thank you, keep learning, stay safe, and keep liking the lecture. Okay, if you like the lecture, do like it, comment on it. If you have any doubts, do ask. Thank you.